Welcome everyone. Here we have started in the previous class, we have started with the new chapter, that is photosynthesis. We have seen the introduction for photosynthesis, that is what is photosynthesis, how light energy is utilized in producing the food material for the living organisms all around the world. Photosynthesis is the only process which provides us with the food, and along with the food it also provides us with the oxygen. And we have seen how those factors are responsible. What are the factors which are responsible for photosynthesis? That is, for photosynthesis to take place, water is essential, chlorophyll is essential, carbon dioxide is essential, and sunlight is essential. These are the four most important factors which are required for the process of photosynthesis. In the absence of even one factor, photosynthesis cannot be performed. We have seen this with the help of this various experiments. Various experiments have proved that in the absence of even one of the factor of photosynthesis, photosynthesis process cannot be performed by the plants. It will fail to perform the process of photosynthesis in the absence of any single factor. Okay, now let us move on to historical, that is contributions of the different scientists for this process of photosynthesis. Here this experiment, here the first scientist, that is the name is Joseph Priestley. Joseph Priestley is the name of the scientist. He discovered oxygen. He discovered oxygen. We have seen this one. We have known this, that Joseph Priestley is the scientist who have discovered the oxygen. Who have discovered oxygen in the 7th class itself, in the 7th class, previous classes. So here now the scientist Joseph Priestley, he stressed on the importance of the air. He stressed, the Joseph Priestley, the scientist, that is by the experiments, the research he did on the experiments which he performed. Here what he said, that he pressed on the role of air in the growth of the plants, how air is essential for the growth of the plants. Okay, so he did his experiment. How he did his experiment, that is what he used. He used a candle, a mouse, and this bell jar. Candle, mouse, and a bell jar is used to perform the experiment. Okay, so in this experiment what he did was, he kept a mouse and a candle, burning candle, inside the bell jar. Okay, it is closed. Afterwards it is closed. So the candle burned for a little minute, that is few minutes, and the mouse was also survived even when it is closed. When bell jar was totally closed, the mouse and candle, they lived only for a little time. Afterwards what happened? The mouse died, and the candle which was burning, it also stopped. The candle also stopped. He did this in the sixth setup. Here he took this bell jar. Okay, closed one, bell jar is taken. So okay, mice was taken here, and candle. He kept the candle burning. The candle was burning. Okay, he kept it set up for some time. Afterwards what happened? The mice which was alive some time before, the mice died, as well as the candle which was burning. The candle which was burning, the candle was also put off. So by this he concluded that there is something, this mouse and this burning candle, what they are doing is, because when the bell jar was closed, air was still there inside the bell jar. When the bell jar, when he closed the bell jar, air was present. So what he deduced from this, that is when his with this finding, that these burning candle and this mice has taken out something from the air which was present inside this bell jar. They have taken something out from the air, and because of which they have damaged the air. They have damaged the air. Who have damaged the air? This mouse and this candle has damaged the air. How they have damaged? They have removed something from the air. They have removed something from the air. So because of which the candle and this mice died. Later what he did? So here later, along with this mice and this candle, he also put a mint plant in this. He also put a mint plant inside this bell jar and kept it. Now three things are there. Plant is there, candle is there, and mice is there. Okay. So what he did? Now after the presence, when the plant was put, mint plant, small mint plant was put, what happened? All the three of them survived. Even when the bell jar is closed, all the three, that is this mice as well as this mice survived, and the candle kept burning. So what he deduced is, so whatever these two has removed from the air, this mint plant has provided it. Mint plant has provided the air, that substance. Mint plant has provided the air, that substance which was removed, previously removed by this candle and this mouse. So what he deduced? What was the thing? What is the plant giving? The plant is giving oxygen. So earlier what they did, air was there, now inside the air oxygen was present. So in order to survive, the oxygen from this air has been removed by this mice for respiration and this candle for burning. Okay, whatever oxygen was there in the air which was in the bell jar, that has been utilized, that has been consumed by this burning candle as well as this mice. So that is why in the absence, now whatever oxygen was there in the air has been utilized by this mice and the candle. There is no further more oxygen. There is no further more oxygen in the air, and in the absence of oxygen, the candle burning cannot take place and the mice also cannot survive. That is why the candle was stopped burning and the mice died. Okay, then what he did? He kept this plant. Now plant, when it performed the process of photosynthesis, it released oxygen. It released something in the air. They have taken the oxygen from the air. Now this plant is releasing that, giving that oxygen back to the air. So continuously plant is giving that oxygen back to the air, and this, because of this oxygen, continuous supply of oxygen, the candle is kept burning and the mice is also surviving. So what he said, that the plants restore to the air. Plants restore means give something to the air. What breathing animal and burning candles remove. So what they removed? They removed this burning candle and the breathing animal, removed the oxygen from the bell jar, and that removed oxygen is restored to the air by the plant. So plant is giving oxygen which was removed by this animal and the candle for burning, and animal by breathing. So that is what he discovered. Along with this, he discovered during this process, he discovered the oxygen. Okay. So now coming to this another scientist whose name is Jan Enginehouse. The name of the scientist is Jan Enginehouse. Enginehouse, he demonstrated that sunlight is essential. He demonstrated that sunlight is essential. What this Joseph Priestley said, that plant is giving certain plant is restoring the substance which has been used up by this breathing animal and the burning candle. And for the plants to restore that substance, plants require sunlight. He said, Joseph Priestley said that whatever has been removed from this bell jar by this mice and the burning candle, that is given, whatever this mice and burning candle is removing from the air, it is given to the air by the plants. Okay, this was said by Priestley. Now here this Enginehouse, he also said the same thing, that is plant is giving the air back something which has been taken up by the mice and the burning candle. But he also said that for the plants to restore that thing which has been utilized by the mice and the burning candle, plants require sunlight. Plants will be giving oxygen to this, because plant, that is these two are taking the oxygen. So plant is giving oxygen to that, and for the oxygen to be produced, for the oxygen to be produced, for the thing to be restored in the air. Plants will be restoring now. So what plants require to produce that oxygen? Plants require sunlight. Plants require sunlight. This was said by Enginehouse. Okay, what Enginehouse said? The plants to restore whatever was damaged from the air, plants require sunlight. So sunlight is also essential. Now another scientist whose name is Julius von Sachs. Another scientist, that is Julius von Sachs. Okay, he provided evidence for the production of starch. He said that starch is produced by the green parts of the plant. Okay, starch is produced during the by the green parts of the plant. What he said, that starch is stored in the green parts of the plant. So now green substance. He also said that there is some green substance which is located in special bodies. He also said that there is some green substance present in the special body of the plant. What is that green substance? That green substance was later identified as chlorophyll. That green substance was later identified as chlorophyll, and he said that this chlorophyll, the green substance, is located in special bodies. So that green substance is chlorophyll, and where this is chlorophyll is located in special body. Later the special body was called as chloroplast. Later that special body was called as chloroplast. So what he said is, the plant will be, that is, there is some green substance in the plant, which that green substance is nothing but chlorophyll. It was later discovered, but that time he just said that there is some green substance in the plant, and that green substance is present in some special structure. Later that special structure is called as chloroplast, and because of this presence of this green substance which is there in this special structure, so plant is able to produce starch, and the green part store starch. He provided that evidence that there is something green which is present in some special structure, and that green and that special structure is responsible for the production of starch, and starch will be stored in the green leaves. This was said by the scientist Julius von Sachs. Another scientist whose name is T.W. Engelmann. T.W. Engelmann, you can pronounce it however you like it. So here another scientist, he used cladophora. He used cladophora for his experiment. This cladophora is nothing but alga, green algae. He used algae for his experiment. Okay, and how he did his experiment? He used this setup. He used this prism, and that using this prism will split light. Using the prism we split light. So that technique is used by the scientist Engelmann. Okay, he used prism, and along with the prism he also used this green algae. By taking those two things he did his experiment. What he said that, so here if I draw this one, if this is the, so he took something. In this now he placed this cladophora, that is green algae. Algae is mostly present in the water, on the water. So that is why that is, just for your understanding, I am saying a beaker was taken, and inside the beaker a little water is there, and here on this alga is placed. Algae is placed on the green water. Now here, because of this taking the prism, the light is split. That is, light is passed through this. When it split into 7, when the light split, that is, we split into VIBGYOR, 7 colors. The light split into the 7 colors, forming this webbed shear. Okay, so what he said that, so when he has taken this cladophora, green algae, with this prism, when the light was split, what he said that bacteria accumulated in blue and red light of split spectrum. Where they? this all those algae was there. Now algae, this cladophora is nothing but blue-green bacteria. We can even call it as blue-green bacteria. So where it accommodated? At the blue and red light. Here the bacteria, all those bacteria accommodated here at this place and at this place. Okay. And some bubbles were released at this place. Some bubbles were released. So that bubbles are nothing but those are the bubbles of oxygen. Those are the bubbles of oxygen. So what he said, Engelmann, he used this cladophora and along with this prism, he said that bacteria is accumulated in blue and red light of split spectrum. So we are splitting this light now in the split spectrum. This is the split spectrum where this bacteria is accumulated. The bacteria was spread here, but all this bacteria, few bacteria is accumulated in this region and few bacteria is accumulated in this region. So this was the first action spectrum. This was the first action spectrum. Okay, so where this bacteria accumulated? In the blue region and the green region. Why they accumulated? Because they have, why they are called as blue-green bacteria? Because they have chlorophyll. Because of the chlorophyll, when they have got the specific light of wavelength, they have performed the process of photosynthesis. So where is photosynthesis? Later this action spectrum gave rise to another spectrum where we said that rate of photosynthesis is more in blue region as well as in red region. We'll see another type of this action spectrum in the coming video, but as of now you just remember this one. Okay, so the action spectrum which was given by this Engelmann, he used cladophora or the blue-green bacteria and the prism, split spectrum. So what he said that bacteria accumulated in the blue region, blue light region and the red light region. Another scientist whose name is, so by this time the scientists that is understood that the process of photosynthesis, for the process of photosynthesis to be taken, that is performed, plants require carbon dioxide and water, and in the presence of sunlight, till these discoveries, now scientists understood that for process of photosynthesis, carbon dioxide and water is utilized in the presence of sunlight, and it produces carbohydrate and releases oxygen. This was clear by this time with all these inventions and the discoveries. Now later another scientist whose name is Van Niel, here Cornelius Van Niel, this is the name of another scientist. He observed that photosynthesis, he did this experiment by taking this blue-green bacteria. He did his experiment by using the same blue-green bacteria. So by taking this blue-green bacteria, he said that photosynthesis is a light dependent reaction. That is, photosynthesis is dependent on the light. In the absence of light, photosynthesis will not be performed. So here by this, when he kept this thing during the sunlight, if it is kept, it will be, that is, during the sunlight, this blue-green bacteria will perform the process of photosynthesis, and in the absence of sunlight, these bacteria cannot perform the process of photosynthesis. So that means sunlight is very essential for the bacteria, blue-green bacteria, to perform the process of photosynthesis. Who said this one? Van Niel. What Van Niel said? First thing which he said is sunlight is very important for the process of photosynthesis. Without sunlight, these bacteria will not perform the process of photosynthesis. The same thing which was said by the scientist. Which scientist here? Enginehouse. He said that sunlight is essential. Okay, so this was again proved by Van Niel that photosynthesis is a light dependent process. Now he also said that earlier it was found that oxygen is released during the process of photosynthesis. Oxygen is released. Previously it was thought that carbon dioxide, that is oxygen from the carbon dioxide is released as oxygen. Oxygen, that is the oxygen which is released during the process of photosynthesis, is taken from the carbon dioxide. Is taken from the carbon dioxide. And hydrogen, that is hydrogen reduces carbon dioxide to carbohydrates. What he said that, for example, here what we said, carbon dioxide is utilized by the plants to perform photosynthesis and produce carbohydrate. That is, if we write this equation, carbon dioxide plus water, it gives carbohydrates, that is C6H12O6. So that means from where this carbons, 12 carbons we are getting, so it is getting this hydrogens which is given to this carbohydrate is from the water. Hydrogens which is given for this formation of carbohydrate is from the water. So water is giving the hydrogen to water is giving the hydrogen to carbon dioxide, and because of which carbohydrates are formed. What is said that, earlier it was said that here this carbon dioxide is helping in the formation of carbohydrates. How it is helping? It is releasing the oxygen and hydrogens is taken by this carbon dioxide, carbon, to form the carbohydrates. It was said by this scientist. Okay, now later, so according to this one, the equation of, this was the equation which was given. This was the equation which was given. Why? Because earlier it was said that hydrogen is taken up by the carbon dioxide to produce carbohydrates and oxygen is released. So the oxygen which is released is from the carbon dioxide. But later what happened is, there were few bacteria, there were few plants, there were few organisms which was not releasing oxygen during photosynthesis. Most of the organisms release oxygen during the process of photosynthesis, but there were some organisms which did not release the oxygen during photosynthesis. So what happened? Why those organisms did not release oxygen? Here oxygen was released. Plants have taken the water and carbon dioxide and oxygen was released, but it was assumed that the oxygen which is released is coming from the carbon dioxide. Okay, so from this carbon dioxide oxygen is released and these hydrogens are added to this carbon to make the carbohydrate. It was assumed, but afterwards later on they found that there are few plants which does not release oxygen. So how they does not release oxygen? Why they does not release oxygen? Why? Because instead of water, that is, they have taken H2S. Instead of H2O, those plants has taken H2S. H2S was taken. So when H2S was taken, the product was not oxygen. The product released was not oxygen. The product was sulfur or sulfate, not oxygen. So that means, so with this they said, okay, the oxygen, we said now oxygen, carbohydrate plus oxygen is released. Earlier it was thought that this oxygen is from the carbon dioxide which is released, and this carbon and hydrogen are joining. This carbon and hydrogen are joining and forming carbohydrates, and oxygen from the carbon dioxide is released as O2. It was earlier thought. Now this plant which take hydrogen sulfide as the source, hydrogen source, it is not releasing oxygen, it is releasing sulfur or sulfate. So that means here in these plants, oxygen is evolved. The oxygen which is evolved is not from the carbon dioxide, it is from the water. It is from the water. Earlier it was assumed that this oxygen is released in the form of, that is, this oxygen is released, the carbon dioxide, the oxygen from the carbon dioxide is released. Now after this it was concluded that the water consists of oxygen, and this oxygen is released, not this oxygen. This oxygen is released in the form of, that is, this oxygen is released. So here this water is used and oxygen is released from the water, not from carbon dioxide. So the source of oxygen that is released is by the water. It is by the splitting of water oxygen is released, not by the splitting of carbon dioxide. So this was said by, so that is, the oxygen comes from H2O, not from carbon dioxide. This was given by Van Niel. Van Niel, he said, by doing the experiment on purple, blue, green bacteria, he said that light is essential. Photosynthesis is a light dependent process, and here oxygen which is released during the process of photosynthesis, it is coming from H2O, by the splitting of this H2O, not by the splitting of this carbon dioxide. So this was all about the different introductions, historic, that is different scientists, works of different scientists on the process of photosynthesis. In the next class, we'll take up with the next topic.